Hello and welcome back to Multibiomics. I hope the videos from the module 1 were in some ways helpful for you to get familiar with the computational analysis part of the single cell RNA data. Now, if you haven't gone through those videos, I would request you to please go through them once, as following a few videos which are coming are a continuation of the first module. And for those who are here for the first time, I would like to tell you that this course also comes with a free subscription to Google Classroom, where you will find all the previous lecture material and the codes, and you can have access to the video links, the link to my GitHub repository, everything for the lifetime. It is free, and you can go at your own pace. And I also encourage all the viewers and participants to ask for doubts and start their own discussion in the Google Classroom platform. The link to join the WhatsApp group and the Google Classroom will be provided in the description box below. Also, feel free to connect with me on LinkedIn if you have got any doubt. If I get time, I'll definitely try to resolve the issue or any doubts that you have. So without any further delay, let us start with the second module. And that's why I once again welcome to this hands-on tutorial on single cell RNA sequencing data analysis using the Seurat package in R. In this session, we will explore key aspect of a Seurat object, focusing on understanding the variability in molecular counts across all the cells present in our dataset. We'll be visualizing gene expression pattern, and we'll be navigating the complex data structure within the Seurat object, right? So we'll start by plotting the distribution of the detected molecules and the genes per cell to assess the data quality and the variability present in it. Then we will dive into the Seurat object itself, and we'll examine the different assays and the different expression matrices, and inspecting the metadata that provides the valuable cell level information. By the end of this module, you'll be comfortable in accessing and interpreting the essential components of your single cell dataset, setting a strong foundation for downstream analysis like clustering and differential expression. So let's get started. But before that, if you have gone through the last video of the first module, we were going on with the clustering and annotation of the PBMC dataset, the single cell dataset. You would remember that we have used at the last part of the script, we have saved the Seurat object with the help of save RDS or save reads function. And we have named it as PBMC underscore 3K final dot R. So it's an R object. What happens is, like, many layers of the information, like important markers associated with it, top 10 variable genes, all those data are not stored because they are not the part of the Seurat object. So in that case, what you can do, you can list all these objects and save them separately, and then you can load them as you need them. But for the sake of this demonstration, what I have done is, like, I have saved the whole image file of the entire workspace, and with the help of the save.image function, I've done that. This will help me to load the entire environment without losing any data. But this step, mind you, is not the best practice when working with a large scale and reproducible data, because it is very memory intensive. As we work further downstream with the analysis, as we proceed, the data, it gets more data intensive, right? So it's better to save what you need separately, and you can load as you go. But for the sake of this demonstration, I have loaded the entire workspace. The changes I have already made in my GitHub repository. But still, if you want to see that, yeah, so here in the single cell analysis classroom, if you go to classwork section, here exploring Seurat object and visualization topic, you click over here, and I have, sorry, not this one, because I'm going to tell you about the previous code, right? So in the first one, an introduction to common analytical workflow, in that I have given you the GitHub R code. I mean the link to the GitHub repository where I have this R code, and you click here and see the code for the module 1 is here. So at last, what I have done is I have used save RDS, right? And I've saved the PBMC object alone. And in this case, I have saved the object as PBMC3k underscore final.rds. I have given this name. But there are many things that I'm losing, like I'm losing these different IDs, right? And I'm also losing the top 10 gene which has the most variability. So these are some things that I'm losing, because these are not the part of my PBMC Seurat object. Okay, and that is why I have added this one. I can what I will do, I'll just edit it. I'm editing it here a few minutes. I'm just commenting it out. So better for sake of this demonstration, to save the entire image file, and I'm saving this as pbmc underscore workspace.r data, right? And I'm saving it. Before saving in the GitHub, if you have an idea, you always need to commit the changes to the main branch itself. The changes committed. So I wanted to demonstrate this. So now moving on with our script. So my environment is empty, nothing is there in my environment. This was the plot generated earlier. This one, it's okay. So you have to set your working directory also. I've already set my working directory. You have to go to session and you have to set the working directory from here, right? Choose directory. Load all the important libraries: Seurat data, ggplot2, patchwork, dplyr, and run to load all these libraries. All these libraries are already loaded. And now what I'm doing, since my environment is empty, I want the previous work from the module 1 to get loaded. I'm loading it. I'm just running this command. The command is load, and you have to start with the bracket, and you have to give the path where you have stored the pbmc underscore workspace.r data, or whatever name that you have given. This is the relative path that I'm giving, right? So the 2 dot means you're going one step behind the working directory. So my working directory, if you can see in the right hand corner here, hg19 folder. So what it will do, it will skip this, it will go back to the filtered gene DC, that is barcode matrices. Okay, and inside that it will look for the output directory. So I have got the output directory here. So if I open the output directory for you, you see the workspace.r data is stored over here. So this, it will load, right? Go back to my working directory and I'll just run this, and within few seconds, see, all things are loaded over here, right? You see, for this first cluster, that is cluster 0, I have got the markers. For the third cluster, cluster 2, I've got the markers associated. I've got the PBMC object.data. Then I've got different markers, right? And various plots are stored. Then the most important one, top 10 variable genes are stored over here. So all these things. So now if I want to start by exploring our data, so we'll be exploring our data with the help of generation of few plots. So if you see in this step, we are using the ggplot function. Okay, in this, first I'll just run and I'll show you that I have generated an histogram. So what have we done in this step? Maximize it first. So in this, we are examining the variability of RNA molecule detection across single cells. Specifically, we use the N-count RNA column from the Seurat object's metadata. So where is the Seurat object's metadata? If I go here, and you can see this is our Seurat object, PBMC was our Seurat object. I've clicked, and here inside that metadata, I have got N count underscore RNA and feature RNA. So the counts of RNA and features and percentage of mitochondrial gene that we have already calculated in our previous module 1, everything is stored in the PBMC object, right? So this PBMC is actually a Seurat object, okay? And everything is the data associated with the a Seurat object, right? So fine. So everything is present over here. So that's what I'll tell you how to explore our data. So going back to the script, we have generated this particular histogram, and let's see. So in the y-axis, we have the number of cells, and in the x-axis, we have the total RNA count. So RNA count are nothing but the unique molecular identifiers, or we also call them as UMIs, and how many UMIs are detected per cell. This idea you'll get with the help of this histogram. Okay, so you can see that more more than 300 or like more than 250 and a little lesser than 300 cells have more than a little over 2000 genes expressed. So as you can also see the distribution of the data, it is slightly skewed toward the left side, and it is a single modal normal distribution, kind of a normal distribution. It's not a bimodal or multimodal data. So you should know about the distribution of your data also, right? Then it is very critical in the early step in quality control to explore your data, because it will help you to flag the low quality or overly high count cells that might skew your downstream analysis. So we are using the ggplot2 to create a clean minimal plot of the histogram. So not only with the help of histogram, but we can also plot the violin plot. If I run this code, we are using the violin plot to examine our data here. So here also total RNA molecules detected per cell we are visualizing, and using the violin plot function, we are focusing on the N count RNA features, which will reflect the total UMI count per cell. And it is a very important metric for assessing the cell quality and the complexity of the library, which was used to prepare the wet lab analysis. The shape of the violin plot will highlight both the distribution and the density of the UMI counts, or the number of the RNA molecules detected per cells. Okay, we are doing it across all the cells. So let me maximize this plot. So each cells over here is represented by dot. So you can see most of the dots are present over here. So naive CD4 T cells. So and you can see each cell, each dot is a different cell, and for like, so you can see over here different colors of the violin plot. All these things were done in module 1, and we have clustered all the cells, and we have got 1 2 3 4 5 6 7 8 9 clustered, and we have colored them differently according to their names. So you can thus go through the module 1 once again, and you can understand that what this data actually represent. And I've also explained how to interpret a violin plot there also, right? So here, with the help of violin plot, you will gain more information as compared to histogram, and you can also detect outliers. If you have a huge number of huge quantity of gene expression, and it might be a doublet cell, that is two cells are captured at a single well or a single droplet, like that. Okay, or you can also plot a scatter plot, and you'll get a very valuable information with the help of the scatter plot. If I run this, we'll get a scatter plot generated. Now, this plot, same plot was generated in the module 1 also, and you could there everything was colored as with with a single color. Each dot were of single color, because this step we had done before clustering. And now, since everything is clustered and all the cells have got their identity assigned, so each dot has different colors, right? Now, coming to the shape of the data, shape of the plot, linear relationship can be seen. Like roughly, it's corresponding to the straight line. So as you can see in the y-axis, it is giving you the number of genes. Feature is nothing but the genes or RNA, and the so it's like gene name. Sorry, the genes that are present, and the count in x-axis. Now, if the number of features are more, right? So number of features, that means the genes. That means the number of RNA molecule will also be more, because genes are nothing but the RNA molecule, and definitely it will be a linear relationship. So this will give an insight whether your data is, properly prepared, during the wet lab workflow. So yeah, we would expect a linear relationship. So again, if you want me to repeat, is like, if the count of the RNA molecules in x-axis is more, then definitely the number of features that we have got should be more. So UMI counts versus the number of genes. So number of genes and UMI counts, they show positive correlation. And yes, we have got that positive correlation with the help of a straight line sloping upwards, right? So like that, we can analyze our data. And moving on to the next section, now we are going to explore the Seurat object. Now, in our Seurat object, which was PBMC, so how I've got this one is like, go to the environment pane in the right hand side and click on the PBMC, which was our, you can click on various objects and you can see how they are looking, right? If I click on top 10, you're getting the top 10, the top variable genes here. Not only 10, it is named as top 10, but we are getting 90 genes. But now we are going to visualize the PBMC Seurat object. And in that, you see the first slot is the assay. Since this is the only assay which has been performed over here and with this data set. So we will be dealing with only the RNA assay. Now, assay actually represents a data slot containing a gene expression or the any other molecules also it can contain, right? For same set of cells. Different assay typically correspond to different data modalities or pre-processing workflow. For example, the RNA assay stores raw or normalized gene expression counts, while we can also store protein assay and other metabolite assay. There are so many. For spatial transcriptomics, we also have another layer where we have matrix for the coordinates where the particular cell is there, and we also have a layer of images, histopathological images. So there are different assays that can be present in a Seurat object. But this is a very simple data set. It has only one assay, which is known as RNA. So you can just click on your inside your environment pane, you can click on the object and you can visualize it. Otherwise, there are many. First, let me close the tabs. Okay, you can also visualize with this function assays. And within the bracket, you have to write the name of the Seurat object, which is PBMC. And if I run over here, you see only one assay, that is RNA, is present. Now, so inside the PBMC RNA, if you want to explore the RNA section, so whatever section that you want to explore, first let us see again what are the different sections present. So inside the assay, we have RNA. And inside the RNA, there are layers, cells, features, default, assay, and all these things are present, right? And we have got metadata and miscellaneous and keys. These are the different things that slots that we have inside the RNA section over here. But you can also run this command. You have to call the Seurat object by the name PBMC, and then whatever you want to explore, you have to put it inside the double square brackets. And then when you run this, so what all the datas we have? We have 13,000. You can look at the console below in the screen, right? So we have 13,714 features. Features are nothing but the number of RNA molecules, number of genes that were detected from 2,638 cells. So usually the gene names are stored a row, and the cells or the different samples are in the column, right? And we have also calculated the top 10 variable features, and they are these genes. These are the gene names, right? And we also have a layer section which we have the counts of the molecules stored and the data, and then we have also scaled the data. So all these things are stored. We can look at them visually, or you can get help from as well. So what are the other slots that are present inside the Seurat object? So you can use the slot names function, and inside that you have to call PBMC, and inside PBMC the RNA slot you have to call and run this function, and you see all the slot names are given: layers, cells, features, default, assay.oric, and metadata, miscellaneous, and key. All these things are given. For example, to look at the expression matrix in the default layer. Now you also have a default layer. So default layer will tell you that inside a particular slot, which layer is actually default one. So let us look at what is present inside layer. So you have explored the RNA slot. Now whatever is inside the RNA, that name also you have understood. So the first one is layers. That is why I'll explore the layers. So the syntax is like same. You have to copy this one and paste, and inside that what you want to explore, layers. So you have to write at the rate, and then you have to put that name. So I'm putting it layers over here. So what's inside the layers? So it's a very big, huge data that stored over, right? See, it's a sparse matrix. This sparse matrix. So 13,714 rows, which is corresponding to the genes, and 2,638 columns, which is corresponding to your cells. So where the values are 0, the numeric values are not stored because that is memory intensive, and each time to read the 0, the memory is wasted. So it's better to put a dot, and that's why it is known as sparse matrix, since there's no numeric value present. Suppose it had 0 instead of this dot, it would be called a dense matrix, and it will be huge in terms of memory, right? That's why mostly we use a sparse matrix only. Now you want to know which is the default section inside RNA. So if you run this one, there's only one default section showing. Let's run this. So inside the layers, inside the RNA slot and layers, which one is the default section? So it will show you. So this matrix, the default thing inside. So most of the functions are worked upon this default field. You can change this default field by yourself also. This code is just to understand what is the default layer inside our slots, right? For this one. So what we have got, so the sparse matrix was the default one. So whatever the operation that we are performing will be performed over this sparse matrix only. Now to check the cells and features name. Okay, now here, so I'm again calling my PBMC Seurat object. Inside I want to I want to get a peek inside the RNA slot, and inside that RNA, I want to look at the cells. If I run this, so the data that is stored here is actually a logical map. So what do you mean by logical map? Logical means it will be either true or either false. So data is stored in this structure, right? And for this, for 2638 values, because there are this many cells and across 3 observations, right? And if I go for RNA features, so there'll be many features, so I'm just counting, I'm just getting head, that means first few lines of these things. So I'm just so 6 values across 3 observations, right? Because head is giving me only 6 values. So what are the 3 observations if you say? So these observations are nothing but what we have observed while working on with the data, and all these observations are stored in the metadata slot. So if I run this, you see the logical map, right? I've told you we have got also the logical map. So these are false, true, all these values of the logical maps are over here. If I want to exactly show what are the 3 observations, is nothing but, so we have counted the mean expression values and the variances of the within the expression values, and the not and the expected variances. So these are the 3 observations, and then later on we have standardized it. But these are the 3 observations that they are counting. Okay, like this you can explore your Seurat object. And then next we have got exploring through visualizations. So I'm planning to upload this as a next part of the video. So this was something which I wanted you to understand, that how you can explore your Seurat object. So anything that you have to call, you have to write the name of the object, and inside that what are the different slots that are present, you have to enclose them within the double big brackets, and if anything is present inside that, you have to call with the help of at the rate function. I hope this was clear, and let's meet again very soon with the next video. Thank you and have a nice day.